Welcome back, ladies and gentlemen. So today we are going to talk about really the crux of why meiosis evolved in the first place. And that's because we want to be able to go through sexual reproduction. In order to do that, we have to ensure that our genetic information isn't doubling every time. And why do we care about any of that? Because this is the way that we get genetic variation into the vast majority of our populations. And that genetic variation is a necessity in order for natural selection to work. There has to be differential success in reproduction and survival, and that all has to be able to occur in molecules that get passed on to their offspring. So, what's that molecule? It's DNA, it's your genetic information. And so, in order for these organisms to get variation that they can pass on, that means we have to recombine our genes. While mutations can definitely be a way to get new variation into our populations, there are a lot of mechanisms in place that have evolved to prevent those mutations from happening in the first place, and when they occur, preventing them from constantly being perpetuated throughout cellular lineages. Because remember, the whole point to genetic information is that it is your set of instructions. So it's really important for cells to have had evolved ways to ensure that their set of instructions was being passed on accurately. So there are proofreading mechanisms in the process of replication itself. There are things like the G2 checkpoint to try and make sure that that genetic information didn't get copied incorrectly before we go through cell division. So, not that those mutations don't slip through, they do, but generally speaking, that's not going to be the bulk of where new variation comes from in a population. Instead, it's going to come from genetic recombination. And that's really just the shuffling of genes around, ensuring that when I produce that offspring isn't a clone of all of my offspring, nor is it a clone of myself or my mate. And so that genetic recombination, that combination of different genes, comes from the independent assortment, the crossing over, those two processes that happen to your genetic information before you pass it on in the process of the production of your gametes via meiosis, and then the random fertilization itself. So either random in the sense that it's a lottery chance which egg and which sperm get fertilized. It's also random fertilization if we have random mating. And so different mating choices and all of that kind of stuff is also random, at least to some level, when we talk about who mates with who, in addition to which of their cells gets fertilized with which of their mate's cells. So independent assortment, I've sort of laid this out here multiple times, but let's make sure we have a clear understanding of what we're talking about. When we look at independent assortment, this really happens during metaphase 1. And it comes as a result of that tetrad formation and the fact that those tetrads then line up on the metaphase plate. And the way in which those homologous pairs orient themselves during this metaphase 1 stage really is random. There is no guarantee to say that all of my paternal information is going to go to the right and all of my maternal information is going to go to the left. They are completely independent of each other depending on how they line up. So if I take a really simplistic example here and just look at a cell with two homologous pairs, I could have this possibility where all of that paternal information does in fact go to one direction, all of the maternal goes the other way. And so I end up with two different combinations there, all paternal or all maternal. Or I could also have them line up like this, where I get chromosome 1 of dad's going this way, chromosome 2 of mom's going this way, and then mom's 1 goes this direction, dad's 2 goes this way. And now I end up with half and half. And so these are also different combinations. And when I look at the organism as a whole, in this case, all 4 of those combinations would be floating around in the gametes that they produce. So when we look at the number of combinations that are possible just from independent assortment, we're talking 2 to the n, and n is whatever the haploid number is that exists in that species. So if we're talking about Humans, we're talking 2 to the 23rd different combinations just from this. Then we can layer on the top of the fact that not only does independent assortment happen, but crossing over is also occurring. And so crossing over happens really early on in prophase 1, where we get the synapse and we get these chimeras that end up forming. So basically, we're going to take those homologous pairs, once they synapse and are connected to each other, they will ultimately end up forming these loops or these X's that you can see here in this electron microscopy image. And those chromosomes then will swap information. And so when we go through and then separate, you can see that this arm of the chromatid stayed all paternal. This one has a combination of both paternal and maternal information. Same here, mostly mom, but a little bit of dad's information got mixed up. It would be the same combination here and here, and then this would all be maternal. So we go through anaphase 2, those sister chromatids split out. And so in this very simplistic diagram, we have a completely paternal cell, a completely maternal cell, and then we have these recombinant chromosomes in these two cells here in the center. And the only reason this works is because, remember, they're homologous to each other. So they're homologous in the sense that the information that I'm swapping here should be the same kind of information I'm swapping over here. So if the eye color gene is here, then the eye color gene should also be here on the same chromosome. Now the sequence for those eye color genes could be different, right? Dad may have given you a brown-eyed gene, mom may have given you a blue-eyed gene, and now that we've swapped that information, we have some new different combinations going on down here based on what was left over in this part of the chromosome compared to what your mom gave you. It's now a little bit of mom, a little bit of dad, and same thing here. The cool thing is, we'll talk about this when we get to genetics, that we can use the percentage of recombination that occurs in offspring between genes to actually figure out how far apart they are on the chromosome. So we can talk about mapping genes based on this crossing over event, how often it occurs, and how far away those genes are really depends on the probability of how likely we are to get genes that separate. So if I've got genes that are really, really close together, likely they'll end up staying together in those offspring. But if they're really far apart on that chromosome, it's more likely that they get separated, which is pretty cool. Okay, so we can add on independent assortment, we can add on crossing over that adds even more genetic recombination, and then we get random fertilization. And even if we don't account for potential random mating, which also occurs in certain organisms, random fertilization is just talking about the same two mates, the same mom and dad, mating together X number of times, but every time they mate, the sperm that they fuse with the egg, what egg and which sperm happen to fertilize is completely random too. So we look at the fusion of two gametes. All right, let's just say they're each with 8.4 million possible chromosome combinations just from independent assortment alone. So we're not even counting crossing over here. And we can produce a zygote with anywhere on the order of 70 trillion different diploid combinations. And again, we're still not even accounting for crossing over. So the amount of variation that comes as a result of this type of reproduction and the processes that go into creating those gametes is just mind-blowing. And that's the reason that we should at least be able to perpetuate the species regardless of the environmental changes that occur, because there's just so much variation that exists. Now, of course, that's dependent on whether or not that variation exists for a particular trait that's necessary, right? There are certainly situations where traits become fixed in a population and everybody has the same characteristic for a particular trait. And also, how quickly does the environment change? How strong is the selective pressure? That can definitely have a major impact on that as well. But there's hope, all right, which is pretty cool. So that evolutionary significance is really huge here. Being able to accumulate that genetic variation favors your ability to survive environmental changes. And that sexual reproduction has a huge, huge benefit above and beyond the cost of the energy and finding the mate, producing the offspring, going through the act of mating. And a lot of these guys also have some level of parental care. All of that outweighs the benefit of the fact that sexual reproduction creates this much genetic variation. And then occasionally you pop in mutations that get us some new variation, and the ability to roll with the punches, if you will, just increases dramatically.